Hello, everyone. And thank you all for joining us today. I hope you can all hear me and you've all entered now our webinar. My name is Anke de Leeuw. I'm a field application engineer at REGENHU, and I'm joined today by my colleague, Mary-Lou Sandry, who is our sales specialist. We're also joined by our co-host for this webinar today, Sumant Bhutoria, the CEO of Alphatech Systems. And together we are excited to invite you to this webinar on the evaluation of bioinks for precision 3D bioprinting of soft transplantable tissues, where our invited speaker today, Dr. Anil Kumar, will share his bioprinting research with us. But before we dive into the science, I would like to start today's session with a brief introduction to bioprinting and also welcome you to REGENHU and see how our company can support your research. So I'll now share some slides with you. Great. So let me briefly introduce REGENHU. So we are a Swiss biotech company founded in 2007 with a mission to create advanced bioprinting technologies that positively impact life science research. We collaborate with a global scientific community who have published more than 600 scientific publications using our instruments. Our focus is on precision, reproducibility, and innovation of bioprinting technologies. I'm sure you all already know this, but I'd like to start off today by introducing what the definition of bioprinting is. So bioprinting is the precise layer-by-layer positioning of biological materials, biochemicals, and living cells used to fabricate functional 3D structures. So it's a tool that allows you to controllably and reproducibly build complex biological systems. Bioprinting allows you to save time by automating fabrication and increasing accuracy of your experiments. By standardizing the results, it dramatically improves the precision, enabling a micrometer level accuracy, which was a major limitation in earlier manual methods. So bioprinting opens the door to patient-specific solutions, allowing you to do personalized medicine, and it can greatly enhance the complexity of the structures that we can now fabricate. Bioprinting has broad applications across biomedical research. The technology can be applied for tissue engineering to create different kinds of organs in the lab. We can also use bioprinting to create tissue and disease models using human cells to create a more relevant model than traditional animal models. It allows for patient-specific tissue engineering, which can enhance drug development and discovery. It's also used in biomaterials research and pharmaceutical applications like tablet printing, where you can develop tablets with defined formulations and controlled release profiles. Using feedback from our community, we've developed bioprinters that grow with your research. So the RGen series bioprinters are modular platforms designed to evolve as your needs change. We have developed Shaper software, which is an intuitive and user-friendly design software that will allow you to plan and manage and design the bioprinting process. The RGen series bioprinters also come with RGen Architect, which is a software that allows you to monitor and change your bioprinting process in real time. So this means that you can change parameters while you're printing. So this can allow you to cut down on protocol development time and increase your print precision and reliability, which gives you greater control over your experiments. Both our tabletop RGen 100 model and the RGen 200, which is enclosed in a biosafety cabinet, offer a range of printing technologies. And as part of our integrated solution, we provide both the tools and the technical and application level support to accelerate your research and discovery. Our platforms are fully modular and allow you to integrate multiple different printing modalities, including direct ink writing, inkjet droplet dispensing, volumetric extrusion, electrospinning, and melt electrowriting. So with REGENHU, you can customize your instrument to meet your research needs. So now that you've seen a quick overview of bioprinting, I'm pleased to now hand over to our co-host, who is, yes, great, Sumant Bhutoria. So Sumant is CEO of Alphatech Systems. He is an expert in 3D bioprinting technologies and our partner and official distributor in India. Sumant holds an MBA and a master's in electrical engineering from Arizona State University, and his company, Alphatech Systems, is India's biggest carrier of 3D bioprinting hardware and our official distributor. So I will now hand over to Sumant, who will introduce our invited speaker, Dr. Anil Kumar, who will show us how we can apply these technologies in a research use case. Thank you, Sumant. Go ahead.
We welcome everyone to this webinar, and it is my pleasure to introduce Professor Anil Kumar, PR, whom I have personally known for a long time. Dr. Anil Kumar is a biologist and expert in cell technologies for biomedical applications, currently leading the division of tissue culture at the Sree Chitra Tirunal Institute for Medical Sciences and Technology. With over 20 years of research experience, his work focuses on cell-material interactions, including cytotoxicity and cytocompatibility, evaluation of biomaterials, 3D bioprinted tissues, and the development of bioreactors for tissue engineering. His lab specializes in the use of primary cell cultures for 3D bioprinting and in vitro toxicity screening, with applications in bone, skin, and eye tissue engineering. To date, he has published over 55 articles in peer-reviewed journals and contributed to 5 chapters and holds 6 patents. So without much ado, I will hand over the mic to Professor Anil Kumar. Sir, please.
Thank you, Sumant, for the introduction. I thank Dr. Anki, Verilu, and then Chitra, if I spell correctly, from REGENHU for giving me this opportunity to share our experience with the REGENHU machine to this wide kind of participants. Can I share my slides? Is the screen visible?
Yes, yes, it is.
Okay, so I'm basically from Sree Chitra Institute for Medical Science and Technology, and from the Biomedical Technology wing of this institute in Trivandrum, which is the capital city of Kerala state in the southern part of India. Yeah. So this is the beautiful campus we are working here. We have a settlement palace here. Long time back, the queen of our Travancore ruled Travancore 16 years from this campus. I mean, from this palace. So it has history also. And this is our new recently built biomedical engineering wing. We have also getting a combinational devices block, which is being commissioned now. So basically, we do technology development in biomedical devices area. So there are various departments in our institute. There are 4 departments in this biomedical technology wing campus. I am from the division of tissue culture. So we all know that biomaterials have evolved through generations. And first generation we can say like it is a inert material. Second generation may be bioactive material, and third generation with living cells and all. So which respond and regenerate tissue. That is the third phase. So whenever we make any biomaterial or a device, we have to refer international guidelines to make sure that whatever we are working on is acceptable to the regulatory agencies. So generally we follow international standards like ISO, ASTM, like that. We adopt ISO 17025 for quality management and implementation in this campus for testing of such devices and biomaterials. And this guideline, 10993 Part 1, give you a brief idea of what tests are to be given, done for a particular device. So based on that, we can choose. In the process of this medical device development and to get translated, there are various stages where we forget to put or we don't include in the research kind of aspects. Generally, we do research, but not aiming at how it can be translated. So certain aspects are there which are required for the translation of any device, medical device that we are thinking of. I'm not going to describe each one of these, but there is a sequence of events and documents that we have to prepare when we think about medical devices. Getting into the topic today, we are talking about regenerating organs, which comes under tissue engineering, where we try to develop biological substitutes. These are functional graphs, practically, which are clinically relevant sizes, which is used to repair, replace, or maintain a damaged tissue function. Now, it's a mix of different disciplines, highly multidisciplinary area, and it contains three basic elements like cells, scaffolds, and the biochemical cues. This is what basically tissue engineering is. I think our audience here is well aware of these technologies, so we don't spend time here. Move ahead of our topic, mainly soft organ engineering. Soft organs or soft tissue engineering strategies try to restore the volume loss. But it is not simply the volume of the organ, but those organs are highly functional, very highly terminally differentiated cells. So when the specificity comes, then the limitations also get added to it. We need cells in very high densities, so that when you use cells for soft organ engineering, the cell survival is a very important aspect. Mechanical challenges, when the tissue is made, it should not collapse. Then how we prepare, how we store, or how we transport these kind of organs once we bioengineer it, it's also a challenge. Then other complications like carcinogenicity, the vasculogenesis, cell death, migration of cells, all comes as part of the general tissue engineering aspect. It's add up to the same here with soft organ cells. This is a simple example where certain organs have been developed in the lab and at least has gone to clinical trials. So this is from the Wake Forest Institute of Regenerative Medicine. Previously, I worked here for this bioprinting aspects. So 3D bioprinting, it's an automated generation of functional products, which is biological origin, which matches to the structural organization of the target organ, where we can make it with biomaterials, aggregates of cells or cells, and then mature it in vitro. This is what generally is three-dimensional bioprinting. When we think about printing, we always consider the ink first. Here, the ink is having living cells into that, so we call it as bioink. So bioink is nothing but a formulation of cells that is suitable to be processed by an automated biofabrication technology. So that's one kind of definition, but generally it is a mix of cells and extracellular matrix with its cues to grow. There are various types of buying: support buying, fidgety buying, structural buying, functional buying. These are all being reviewed and discussed in literature, so we don't get into those. But we are talking about the structural and functional buyings. Whenever we think about 3D bioprinting and considering Bioink to formulate a Bioink for 3D bioprinting, we should consider the general biocompatibility evaluation of biomaterials. Irrespective of the class, the biomaterials or the bioink which is used for bioprinting has to go through certain tests. Basically, the biocompatibility test, which confirms that it is non-toxic and it degrades inside the body and it helps cell attachment. These are the tests which we have to ensure. And it has the mechanical properties that of the target organs. And the ink should be printable with a commercial printer. We cannot make a printer for our ink, but rather we choose an ink which can be printed, which can be printed. And once it is printed, we have to check the stability of the shape and the structure, because we are trying to print based on a computer design. So it has to easily and accurately translate that design into a prototype. And finally, it should be clinically proven. These cells which is added to a bioink is also very important. What type of cell we add to the bioink and at what density. The cell concentration is very, very important. The bioink requires 10 raised to 9 to 10 raised to 10 scale of cells as the density, and we can use functional adult terminal differentiated cells or adult stem cells, or the stem cells that are differentiated, and then we use all of these to try in our research. When we consider about 3D bioprinting, there are various kinds of technologies available, types, and extrusion bioprinting is very common. And extrusion bioprinting print by extruding filaments, strands or struts. It mainly by compressed air or a screw. And the bioink will be extruded through a nozzle or a needle, and a platform will collect those extruded material, and the printer moves in a layer by layer fashion to deposit the ink. So when we think about bioinks, there is sometimes it is misconceived that the scaffold printing is also bioink. No, but we have to clearly say that bioinks are different from inks that doesn't have living component into this. So those inks which contain cells or aggregate of cells or extracellular matrix or biological origin, such kind of ink we can call it as bioink, whereas those material which contains a biomaterial alone, and then we print it and then we add cells later, such kind of inks we can call it as a biomaterial. As I said, there are various printing technologies, each having its own advantages and disadvantages. Extrusion, for example, extrusion printing have a good advantage of high cell density and high viscous binding, whereas its printing speed is a bit compromised, and the resolution also is depending upon the nozzle that you use for the printing. At the same time, inkjet printing, it can print even cell suspension, but it should be very low viscous solution. So these are the advantages, and depending upon the tissue that we are going to fabricate, we have to choose an appropriate printing technology. And our bioink should be tuned or evaluated for that particular printer or the printing technology. Now, when you think about 3D printing, there is a term coming in, 3D printability, which has been described in literature in various ways. Sometimes it is described, 3D printability is described as the properties of an ink, whereas it is also said that it is the relationship between the bioink and the substrate that is used. In other literature, it comes with different parameters and give a good outcome. So the printability is described or defined, explained in different ways. So we can call printability as the ability of a material, when subjected to certain printing conditions, it print in such a way that the outcome is suitable for a desirable application. I think this gives a good complete information of 3D printability. Now, as it was said before, it's different parameters play in 3D bioprinting. The speed with which the print head moves, the nozzle diameter, the nozzle substrate height, then the shear thinning property of the bioink, which we will describe later, the temperature at which it is printed, and the pressure with which it is extruded. These are the very important parameters that one would consider to when you formulate a bioink. The printability of different technology is also different. When extrusion bioprinting is considered, then the extrudability is the main key factor. Whereas in soft lithographic kind of 3D bioprinting, the rheological and the optical requirements are the key factors. So the 3D printability it varies with technologies also. It is not with the ink. So we can say that 3D printability is half from the ink, half from the printer. Because we can evaluate 3D printability only after printing. So when you consider extrusion bioprinting, there are four very key parameters. They are the layer height, print speed, extrusion rate, and the diameter of the nozzle. When the very simple example, when the nozzle is too high, the ink will be dispensed in the air. Whereas when it is too low, it cannot dispense, so that it breaks on the substrate. So an ideal height would be a nice filament forming on the substrate. So we should consider right away from how it starts extruding, where it shows the shear thinning property, how it forms a filament from a drop. When a filament is formed, how it is retained as a filament in a multi-layer construct, and then how it is dispensed. It's a smooth filament or a rugged filament. There is also another point which we call as filament fusion due to intermolecular attraction between these ink molecules. If the molecules of the ink is attractive too much with each other, it won't dispense. It will like to cling together. I mean, I think I have a video for this. So this you can see, when it is intermolecular attraction is more, it will stick together. Whereas when it is reduced, it goes like a filament through filament. This is what from drop to filament formation, what we were describing. Now, when a design is there, the printer should translate this design. And I won't repeat, but I would like to say here, we have the REGENHU machine here, which all the work which I am going to present is from the REGENHU 3D discovery printer. So the bioink will be evaluated in a single layer in different patterns, and multi-layers in different layers. So two-dimensionally and three-dimensionally, the ink has to be evaluated. And the strength of the filament is also very important. We use filament collapse test for that. Depending upon the intermolecular attraction, the dispense of the ink is also varying. We can quantitatively measure the printability by applying some formula. I mean, this Bioink is the first report which shows that printability can be measured by image analysis of two-layered construct. It's a very nice concept. And when we say that printability is not mechanical alone, because our bioink has cells, and the pressure that we apply and the extruding process itself should not affect the viability or the functionality of the cell. So basically, depending upon the type of nozzle we use, the viability varies. For example, a plastic tapered nozzle will give a nice high cell viability, whereas when it is narrow nozzle, metal nozzle, it might affect the cell viability. Again, it also depends upon the type of cell which you use in these two nozzles. My point here is, we have to know how the ink goes through the nozzle to check the viability or ensure the viability of the printed construct. So when we think about formulating a very ideal bioink, in quotes, there are points that we have to check before printing and during printing and after printing. So where the bioink will be stored, how its solubility is, whether it is extrudable, whether it shows shear thinning property, or is it biocompatible. This one we check before we start printing. And once we think about 3D printing, whether it is 3D printable, whether ambient temperature printing is feasible, or the cross-linking process is friendly with the cells, which is in the bioink, and once it is printed, whether it is enzyme digestible or not, and whether the ink supports organization of tissue once it is printed. So tissue maturation is one of the aspects. We started this project in 2016 with the aim of developing hepatotoxicity in vitro test platforms. So the drugs that is tested now and which is available has 90% failure. It is because of the interspecies differences between human and mice, because all these drugs are tested in rodents or rat rodents, basically the mice and the rats. So there is a huge gap between these two. Suppose if we use human cells in vitro, we can predict, not confirmatory, but we can predict that some formulation might go wrong in humans. Then the question comes when we can an option for cells and testing. Why we need to have a tree? The requirement is the hepatocytes, when isolated from the liver, loses its function very fast, as early as 4 hours in the culture medium. So whoever try to culture hepatocyte try to extend the functionality and viability of those cells. So this is why we require 3D bioprint. This is the REGENHU 3D discovery machine we have here. We call it as tissue and organ printing printer because it has 4 print head. I know that there are more now in the new models. It has an microvalve jetting as well as high viscous extrusion head plus melt extrusion. We are trying to develop soft tissues. It's very complex to print soft tissues. Also, we are trying to print liver constructs. Liver is a very complex organ, we know that. So when we started working on this project, we started by developing Bioink for printing. When we started working on Bioink, we considered different phases, how we achieve that formulation. So the bottom, which I have given certain names in the text, is the test that we have done, analysis experiments we have done, and the cartoon shows the flow of our Bioink development program. Very simply, briefly, if I say, we first evaluated physicochemical properties, then in vitro compatibility. Then without cell, the ink was tested in vivo for biocompatibility evaluation, and then 3D printed organ. Then this is I won't spend time here. This is the synthesis of Gelma. We choose Gelma as our biomaterial for including our Bioink, and we have optimized the synthesis process to get an efficiency of 64%. We characterized by NMR. The the specific functional groups were identified and characterized. We checked, the rheological properties, and we modified the formulation in such a way that it can be printed at room temperature. And when I say room temperature, it is 19 to 21 degrees Celsius. When JELMA is used for printing, we have to cross-link the JELMA after printing to stabilize it. Otherwise, when we add medium, it will dissolve again. So we have to cross-link in the presence of photoinitiator, which is and UV, ultraviolet rays, and this might affect the cell viability. So what we have done is for our printing liver construct, we go for very large volumes of construct, which increases the UV time. So we added a photoprotective agent to that to protect the cells from the UV. So we have evaluated this publication is there. So we have made a formulation which will safeguard the cells while UV crossing. Now, once we have this Bioink prepared or synthesized, then we have to go for the hydrogel degradation study. Know how fast it degrades and how much in vitro and in vivo, but in vitro is essential. Whether the material after printing is porous, whether the pore size distribution is accurate or adequate. We have here 150 plus or minus 15 micron when we analyze with micro CT. And once this physical characterization is done, then we think about for a transplantable aspect. When you go for transplanting a 3D bioprinted tissues and organs, we have to refer international guidelines. We have here checked the ISO guidelines, especially 1456. So this is the workflow where design is there, which will be fed into the REGENHU machine, and then it is printed. It is cross-linked. Our printer has inbuilt UV, which will cross-link the printed construct. And we have noticed that over days the construct doesn't swell, it doesn't shrink, it keeps almost the same size. After that, long duration only there, if at all there is a change, and there is not much of weight loss. Once it is printed, after printing with cells also, the porosity has to be ensured. So it shows the porosity after printing. It is a scanning electron microscope image of this construct. It should be repeatable. If I print like 24, 48 constructs, the first construct and the last construct would look same, and the number of cells should be same in all constructs. We also checked the cryostability of the JELMA. We freeze it to minus 80 degrees Celsius, and then we thawed it and checked whether it is stable, and it is stable. Next, we went for the cell addition property. We started using the ink with cells. So we coated the JELMA on plate and seeded the cells, and it adhered and it grew on the surface, which shows that it is supporting cell addition and growth. Then it was encapsulated in the JELMA. Cells are encapsulated, mixed within the JELMA, cross-linked, and checked the viability. It is viable. And then on a construct, we have seeded endothelial cells and try to see whether the cell migrates into the scaffold. And we saw that endothelial cells can migrate into the construct. Then this right side image shows the porosity after printing. You can see high cell density here in this diagram. Now we optimize the cells also. You can see here the construct which histological section H&E staining shows similar to that of a normal rat liver, and the 3D bioprinted construct is functional also by secretion of albumin and urine. This not working. Okay, so this is the printing process. The printing process. It's not going. So once we print the construct, we have to do post-printing analysis like self-proliferation, function analysis like albumin and urea. So once we confirm that the printed construct are viable and it is stable in vitro and it has liver specific function, we check the metabolic ability of that tissue, because this is for hepatotoxicity test system. That means we have to expose these construct to drugs. Once you expose such construct to drugs, the drug has to be metabolized. Now here comes, this slide shows the importance of 3D bioprinting in drug screening. The monolayer, which is shown here, doesn't have cytochrome 3450 3A4, whereas it is less minimal, whereas once it is exposed to a 3D bioprinted construct, the activity is more. That means 3D bioprinted is always better than a monolayer. There are two products from this project. We have a Bioink formulation and in vitro hepatotoxicity test system. Hepatotoxicity test system is now we offer to customers from the outside and inside the campus. And the Bioink is the second technology which has translated pretty earlier than the in vitro construct. So once we optimize the protocol to synthesize the Bioink, we have scaled up to many five times, and we have transferred the technology to an industry based in Kochi, Sire Science. So we have an MOU, technology transfer and technology documents for this, and then we also developed the packing and sterilization methods for this. So it was translated to industry and it was commercialized in 2020. This is the property of the Bioink, self-friendly, and it's non-cytotoxic. Its photoconsumer fluidity can be adjusted by concentration. It's porous. You can print in room temperature. A lot of properties which we evaluate each one of the aspects, and then we transfer. So getting into the in vivo aspect, we first check the hemocompatibility of the printed construct, and we found that it is not hemolytic. Then we go for cytotoxicity evaluation, where we check the cells, the material is safe with cells, or the elutant of the material is non-toxic, and we found that it is non-toxic. Then the cell viability after printing, as well as the cell activity and the proliferation. We have to see that the cells proliferate inside the construct. Then we printed the construct without cells and then implanted subcutaneously in animals, and we found that there are neovascularization around the construct, as well as we found some immune cells, which is very natural. So this is the liver, which is very complex structure. Now we have to mimic that is difficult. So we try to see whether these kind of structures are available here in our construct. So we first tested without cells, again without cells, not inside the skin but in the liver itself. So in the liver tissue, we couldn't see any immune response. There is no kind of inflammation around the implant, as well as we could see endothelial cells in the construct, in the implant site. So that shows that it is vascularized. So this is the roadmap that we are planning to go ahead with. So once we think about bioprinting, and then we have to translate, we have to have done it in a GMP facility, and we are working on those aspects. We are having a good facility in the new block. Thank you. This is my team and our management, as well as my collaborators, funding agencies. Thank you very much.
Please join me all in thanking Dr. Anil Kumar for this very insightful presentation. Thank you for taking us through this journey on evaluating bioinks and then different techniques on how to do printing, and then eventually the huge translational aspect and all the regulatory requirements involved. So it was great to see, and thank you for this great presentation. For our audience members, please address your questions and comments using the Q&A tab. You can also use the Q&A function to upvote the questions of others. And now, while everyone is writing in their questions, I would take just a few minutes to continue on the next step of innovations from our side, from REGENHU, before we go back to open the Q&A section with Dr. Kumar. So, we'll take a short moment.
Yes.
Thank you. Great. So we saw a little bit of how the printing technologies can be applied to extrude these really great structures and how they're translatable. But now I would like to invite you to join us on the journey on what new technologies we've released, which may be helpful for future translation. So specifically, trying to overcome some of these hurdles in biofabricating transplantable tissues. So I will not spend too much time here, since Dr. Kumar gave us such a great background already on some of the challenges to do with printing organs and tissues, also for in vitro. So let's just refresh a little bit on the challenges. If we take a patient who's either undergone a trauma or suffers from disease, the native organ is not performing its function anymore. So at this stage, a patient may need a transplant. However, donor organ availability is very limited, and even when they're available, they may elicit an immune response due to the presence of allogeneic cells and proteins. So this is where bioprinting can offer a solution. So by enabling in vitro fabrication of tissue constructs, we can bypass the need for having a donor in the first place. So it allows you to work with patients' own cells, so using autologous cells to reduce the risk of rejection. And this is where you can kind of overcome this immune response, specifically when we think about transplanting tissues, or so if you think about like soft tissues, liver, or also hard tissues like tendons or bones. At the moment, you get a donor from donor tissue, but think about being able to print the patient's own cells here. So another major challenge is the spatial organization of the different cells and the materials, which is crucial for the function of the organ tissue. This is where bioprinting can provide a micrometer level control, which allows us to precisely position the cells and materials and allow them to interact and interface in 3 dimensions. Then we have the native tissue complexity. So if you think about the mechanical properties, the biochemical environment, and the actual tissue architecture itself, this is where, as we heard today in our talk from Dr. Kumar, where you can carefully select your biomaterials, choose something that's very printable, but also elicits the right response from the cells. And this will allow you to design something that better mimics the properties of the native tissue. When we think about critical size defects, so something where the tissue damage exceeds the body's natural healing capacity, but also of full organs that may need to be transplanted, we start to think about, okay, we need to have quite a large scale design. And this is now limited by the diffusion limit, where cells inside your core are going to become necrotic. So this is where we can use bioprinting to engineer vascularization and create larger, more viable tissue constructs beyond the diffusion limit. So this is where we've now developed a new tool which can help you to build functional tissue architecture that supports vascularization, complexity, and viability. So our latest innovation is the dynamic formulation module. So this is a multi-material microfluidic printhead that allows you to edit your Bioink in real time. So as you can see here in the image on the top left corner, you can plug in 4 different materials into these cartridges. The material then flows through the tubing and goes here directly into a microfluidic chip to then be extruded through a shared nozzle. We have 3 different chips available. So the technology allows on-the-fly mixing of materials, patterning of different material gradients, and also core shell fiber extrusion. So this opens up a wide range of applications, like enabling in situ cross-linking. You can imagine introducing mechanical or biochemical gradients, for example, to mimic the stiffness across a tendon-bone interface. You can create large-scale vascularized 3D co-cultures with enhanced viability and function. So the take-home message here is the DFM will allow you to engineer complex functional interfaces. Now I'll show you a nice video of the DFM in action. We can print these gradients. So what happens is the material will enter the microfluidic chip and then allow you to print a continuous gradient from one material to the next. So you can imagine adding different kinds of cells. You can switch kind of on the fly the materials that you print, but also, for instance, the densities of the cells. So this allows you to more closely resemble native tissues. Then in addition to the multi-material printhead, you can also use core shell printing. So let's see if you're able to view this now. So here you can see our coaxial microfluidic chip in action. So what we're doing is we're printing in one of the cartridges we have alginate, and in the other cartridge we have calcium chloride. So the idea is that we have a shell of alginate with a sacrificial inner tube of calcium chloride, and it's then being directly cross-linked in a bath that contains additional calcium chloride. So as you can see, the method is producing this very long and continuous hollow filament, which is resembling the geometry of a vascular-like channel. So this is kind of one of the other methods, in addition to gradients, where we can use the printhead to create more complex structures. Then I will show you now in the second video on the right-hand side, we're able to perfuse the structure. So you can see here the red dye passing through what we've just printed. So we can see that the lumen remains open and functional, which is allowing the fluids to pass through freely. And you can imagine a bit how this technology could be used to create larger scale perfusible and vascularized tissue constructs. So this is just a quick overview of the new technologies that we've developed here. And for all your research that demands precision, reproducibility, and flexibility, our team here at REGENHU is ready to support you, whether you're developing new bioinks, engineering disease models, or working towards transplantable tissues. Our field application engineering team is here to collaborate with you. So with this, I'd like to thank you for your attention. If you have any questions, please post them in the Q&A window, or feel free to reach out to us via email at info@regenhu.com, or you can connect with us on LinkedIn. So I hope now you've had the chance to type your questions into the Q&A box. And with this, I would like to welcome back our scientific speaker, Dr. Anil Kumar, as we open today's Q&A session. Great.
Yeah, I'm here.
Fantastic. So I can see that we already have a question in our Q&A. So Amrutha has asked, how can vascularization be introduced into the scaffold's organs in terms of mimicking the dynamic environment for the desired applications?
Usual, great, important question. So see, we don't create blood vessels. We create environment for the blood vessel, post blood vessel to come in. There are various ways that a tissue engineered scaffold can be vascularized. We plan like this. So our biomaterial used in the Bioink has all the materials necessary to invite. Because I didn't show that slide, when we implanted the liver, we could see sinusoidal structures inside the construct. So that is the tissue formation inside. And sinusoids are the spaces where the blood is filled in the liver. So this is the answer. We have a material which will help in the vascularization by the host itself.
Great. Thank you for this question. While we wait for more questions to appear, don't be shy. You can use the Q&A box. I would have a question as well. So, Dr. Kumar, in your opinion, what is the greatest challenge in printing translatable soft tissues? You've taken us now on this journey, but what would you point as the biggest challenge?
There are many. The primary one would be the cell number and the viscosity of the ink. Because generally, we start research by buying a printer. That's how one starts research. So when we do that, so that is why we went only, specifically for REGENHU, because we have different printing technologies in one printer. We don't know how the tissue is going to behave when we use an ink in that. So we have like low viscous to high viscous printing capability, so that we can fine tune to our tissue. So one is the viscosity of the ink as well as the density of the cell. For example, if I quote our own experience on liver, the hepatocytes are very large cells when we isolate from the liver. So, say for example, if I want to make 5 ml of Bioink, and I add like 20 million cells to that, my ink volume will be 7 ml. It's because the cell packed volume itself will take some space. So one has to play with the volume, the concentration of the ink, and the viscosity. At the same time, the Bioink property should not change. So these are the challenges. The concentration of the cell, the viscosity of the Bioink, and how you mix it. I think the rest are similar to all other things. Yeah.
Great. Thank you. Yes, absolutely. That was very detailed. I see that we have another question from our audience. So Payman Rusgaard, I apologize for my pronunciation here, has asked us, do you think polysaccharides such as cellulose can be used as a Bioink alternative to GELMA because they are cheaper?
It can be used not because it is cheaper, because one requirement for any Bioink which is intended to be printed and implanted inside the host should be degrading, because that, and the tissue has to be replaced with that, because whatever printing technologies that we are talking about is for soft tissue and soft objects. Ultimately what we need is the host cells has to replace what we have given. It has to eliminate the cells. It has to place its own tissues there. Why? Because by definition itself, tissue engineering means we try to repair, replace, or maintain the tissue function. Our aim is only to that. To meet that requirement, we need to have a material which is degrading inside the body. Suppose the cellulose which is used can be degraded. I'm not sure what kind of length and thing it is said. Sure, it can be, but only thing is we have to make sure that it degrades inside.
Great, thank you. I see we have some more questions appearing now in our Q&A box. So Harish has asked, hello sir, you have presented data on cell viability. Could you please clarify how the assessment was performed? Specifically, was it compared with a 2D culture model? If so, how can the results be correlated with those obtained from a 3D model? And then also in this context, should we be comparing our findings with results obtained using commercially available bioinks in order to validate the performance of our own designed bioink?
Oh, I think there are three parts. So one, when we started this project, we always try to put data why we need 3D bioprinting. We could have used 2D first. So our comparison was at those days was with 2D monolayer. And we know that the cells in the 3D doesn't multiply like that when the 2D culture multiply very fast. So we, based on the ratio, because it is in 3 dimensions, we have to take that number of cells in the petri dish. So we correlated that and we compared always our 3D construct with the 2D. And those data I didn't show because very old data. So, but we have done that. So that is what that was our correlation. But later on, we went to compare the function. Not this kind of viability and all, because that scope was defined. Then we went for the biotransformation ability and the cytochrome for the enzyme activity. That means the printed construct should have metabolically active enzyme to biotransform the drugs that we give to for use in the drug screening purpose. So later on we went to those kind of aspects. So that is for first question. And second, should we compare it with the commercially available other gel? Of course, you can use that, but we are not behind brands, but we are behind, clinically proven material. For example, when we transplanted, we try to use a clinically used sample. It need not be gelma because there may not be a clinically used gelma now. So what we do is we use a material, which is a collagen, which is a sponge, which is clinically used, and then compare it with that. And say that, okay, it is clinically used, behave like that, the printed germ also behave. So that is the second part. I lost the third one. Can you read the last?
Certainly. So the last one, I think you covered it all. So 2D versus 3D, and then comparison to commercially available inks. So then I will move on to the next question. Matti Kesti has asked, how do you intend to sterilize the bioprinting environment and consumables in the GMP production? Is this method already in use in your R&D?
Yes, we follow our own methods. So we are setting up a new facility where it is a biosafety level 1 facility. In fact, we will upgrade it to 2 and further. So how we sterilize our buying is by ETO sterilization. So we remove all the water content, make it completely dry, and then sterilize by ETO gas, ethylene oxide gas. So in our commercial partner, what they do is they give you Bioink in a usable form. That means you can add culture medium to that bottle. It will dissolve when you incubate it, incubate at 37, and then mix the cell pellet into that, directly load it into the cartridge and print. You need not prepare your ink at all. It is given as a formulation which you can print straight away by adding cells. So sterilization is the before the mixing part. So we do by ETO sterilization.
Great, thank you. I think we have time for one final question. So from Likita, we have, thank you for the great talk, Dr. Anil. Do you see AI or machine learning playing a role in optimizing pattern design or print fidelity in the near future?
Oh yeah, yes, sure. Because I cannot say it openly because I don't know much about it, but I have heard that through our connections that AI coming for designing will help you to design the most appropriate CAD model for your organ. So that is one aspect. Now the next is the printer itself is controlled by a AI controlled software, so that it helps you to have an uninterrupted, error-free printing. For example, in our region, very old machine now, we cannot intervene in between in our model which we have. I know that the recent model have other facilities. Because it is having a positive aspect also. Once you start printing, you cannot intervene. It should finish completely without any change perfectly. So that came as an advantage to us because we optimize our Bioink so that we need not touch the printer again once we give a print. But sometimes when you evolve through your Bioink development, you might need to intervene in between to optimize the printing process or the parameters. Now AI can help there in getting the parameters easily done for you. So these are the two areas where AI can come. I think what I know. Oh, 3D bioprinting is applicable for single cell culture. There is another question. Can I answer that?
I think we have time. Yeah. Last question. Sounds good.
Yeah. Yes. But in what context it is single cell culture, I couldn't understand. But there are printers in the REGENHU itself having a micro valve extrusion method, and the just one dynamic fabrication one. That also I think in low viscous inks, if I am correct, but I understand this. So in such cases, definitely, if for example cartilage, you should have less number of cells, more amount of extracellular matter. So it is possible. It depends upon the tissue. Yeah.
Great. So I think we've reached our allotted time now. I would like to thank everyone for this great discussion and active participation in our webinar today. Please join me in once again thanking our speaker, Dr. Anil Kumar, for his excellent presentation. If anyone still has any questions, please feel free to reach out to us via email afterwards. We'll make sure that your questions are all answered. Then I would like to thank you all for attending our webinar today. Thank you to our co-host Sumant. And I would like to wish you all a great afternoon and a wonderful weekend ahead. Thank you.
Thank you very much.